Welcome to the part 2 of our lecture on histamine. For the outline of this presentation, the first part of the presentation will talk about the physiologic roles of histamine. We are also going to discuss about the pathological roles of histamine, its pharmacological effects, the mechanism of action, and the therapeutic uses of histamine. For the learning objectives, this presentation aims to identify and describe the pathophysiologic roles of histamine. It also aims to identify and describe the pharmacological effects of histamine and describe the mechanism of action, MOA, and the therapeutic uses, if any, of histamine. The endogenous histamine has both physiological as well as pathological roles. The first part of this presentation will talk about the physiologic roles of histamine. These are the physiological roles of histamine. So histamine has a role in neurotransmission. It is a neurotransmitter in various brain areas involved in activating the sensory nerves, resulting in pain and its sensations. It has also a role in the CNS. For example, it regulates the body temperature, CV function or the cardiovascular function, and the regulation of the anterior pituitary hormone release. It has also a role in tissue growth and repair. It is also important for the stimulation of the gastric acid secretion. So histamine is considered to be the most important regulator of the gastric acid secretion, and it stimulates the secretion via the H2 receptors. It has also a role in the regulation of the microcirculation in tissues. So histamine relaxes the arterioles and increases the capillary permeability. It also regulates the intestinal motility. This diagram shows the major physiologic actions of histamine, its clinical manifestations, and the receptor subtype involved. In the lungs, the main physiologic effect of histamine is bronchoconstriction, and this is manifested clinically as asthma-like symptoms, and it is mediated via the H1 receptor. For the vascular smooth muscle, it induces dilation of the postcapillary venule and the terminal arteriole, and venoconstriction. It is manifested clinically as erythema, and this is mediated via the H1 receptor. For the vascular endothelium, it induces contraction and separation of the endothelial cells. It is manifested clinically by edema and the wheal response, and it is mediated via the H1 receptor. For the peripheral nerves, its main effect is sensitization of the afferent nerve terminals. It is manifested clinically as itch and pain through the H1 receptors. In the heart, it causes minor increase in the contractility as well as in the heart rate, and it is mediated via the H2 receptor. In the stomach, there is an increased gastric acid secretion that is clinically manifested in peptic ulcer disease and heartburn, and this is mediated also via the H2 receptors. So in the CNS, the major physiologic action of histamine is that it acts as a neurotransmitter, and this is manifested by circadian rhythm and wakefulness, and this is mediated via the H3 receptors. So the next part are the pathological roles of histamine. So when we say pathology, it refers to the study of the essential nature of diseases. So these functions are related to the role of histamine in some diseases. So histamine is important in the development of allergic reactions and anaphylactic shock. The binding of antigenic substances to IgE molecules on mast cells causes the release of histamine. Histamine is important in certain cases of hypotensive crisis, increased vascular permeability, intense constriction of the bronchi and the bronchioles, which collectively constitute histamine shock. Another important pathological role of histamine is that it is important for the genesis of peptic ulcer. So histamine can induce hypergastric acidity. Histamine has also an important role in inflammation. So it may be involved in the vasodilation observed in the inflammatory process. Other pathological roles of histamine are: it can be important in the genesis of headache, and it has also a central role in vomission. It is also important in the development of intense skin itching and infections and parasitic infestations. Histamine has a central role in the development of certain allergies and anaphylaxis. Before we are going to define what is anaphylaxis, we first need to differentiate it from the usual allergies. When we say anaphylaxis, that is also referred to as the severe allergic reaction. It involves an individual's breathing and or circulation. Anaphylaxis is considered to be the most severe form of an allergic reaction and is life-threatening. An important aspect of allergy and anaphylaxis management is prevention by avoiding the cause. But what is anaphylactic shock? So it occurs due to the introduction of an allergen in the body. This leads to mast cells or basophils to release histamine and other chemicals system-wide. So this will decrease the tissue perfusion, causing shock to occur. How does an allergen enter the body to cause anaphylactic shock? So an allergen can enter various ways, such as via the injection, inhalation, oral, or contact with the skin. Known substances that can cause anaphylactic shock include food, such as shellfish, peanuts, eggs, and milk. We also have medications, vaccines, contrast dye, NSAIDs, antibiotics such as penicillin. We also have insect venom, physical exercise, and a known cause that are referred to as the idiopathic causes. So anaphylactic shock can occur either due to an immune response, where IgE antibodies are created, or due to a non-immune response. Regardless of the reaction, both cause the same signs and symptoms and occur because mast cells or basophils release histamine and other mediators of inflammation. For the IgE-related anaphylactic reaction, this involves a patient that has to experience sensitization for reaction to occur. Sensitization is where the patient has a first-time exposure to an allergen that causes the creation of the IgE antibodies. So in this diagram, we have here the presence of an allergen that will serve as the antigen. This particular allergen will trigger the production of the IgE antibodies by the B cells. Then, the antibodies will attach to the mast cells or basophil and hang out waiting for the second exposure. So in this diagram, we have here the B cells that have produced now the antibody IgE in response to the presence of the antigen, which is an allergen. After the production of the B cell, after the B cells produce the IgE, So the IgE will now bind to the mast cell or the basophil. Then, when the second exposure to the allergen occurs, it will cause the mast cells or basophils to release histamine and other substances. So we have here the presence of the subsequent exposure to the same antigen. This will again trigger the bridges. Now, antigen bridges the gap between the two antibody molecules, and it will cause the degranulation of the mast cell or the basophil and release histamine and other mediators. After the histamine will be released, histamine will now act on different tissues, such as the microcirculation, to increase the permeability and extension of the blood capillaries. So all of these events will lead to the development of the anaphylactic reaction. It is a type 1 hypersensitivity reaction. This means that the allergen attaches to immunoglobulin IgE antibodies, and these antibodies are created due to this particular allergen. So this leads to a system-wide release of histamine and other mediators, and these effects constitute the anaphylactic shock or the anaphylactic reaction. So after this histamine will be released in the circulation, it will act on different receptors. For example, on the blood vessel. So in the blood vessel, particularly on the arterioles and the capillaries, it will induce dilation of the blood vessels, lowering the blood pressure and tissue perfusion. It will also induce bronchoconstriction, narrowing the airways and respiratory failure. In the heart, it will result to increase in the heart rate. It will also increase the permeability of the vessels, leading to swelling and depletes the intravascular space of fluid that shifts to the interstitial space. It will also induce itching, increasing the contraction of the GI stomach muscles, and increases the gastric secretion. So we have here another diagram showing the important steps as well as the major cells involved in the process of the allergic reaction. So we have here the presence of the first allergen exposure. This allergen will trigger the plasma cells to release IgE. So in first exposure to the allergen, the IgE will just attach to the surface of the mast cells, and upon subsequent exposure to the same allergen or the same antigen, it will cause these mast cells to degranulate, releasing the allergic mediators such as histamine. Constant exposure to the allergen could lead to chronic allergic reaction. The next slide will talk about the allergic reaction that is common in humans, and that is the cellular mechanism involved in peanut allergy. A peanut allergy appears early in life and tends to persist indefinitely when compared to other food allergies. The symptoms range from urticaria to severe systemic anaphylaxis. So different proteins are associated with these clinical reactions. The ARA-H1 protein is an important allergen and is recognized by 90% of peanut-sensitive persons. This figure shows the illustration of the major peanut allergen ARA-H1. So this picture represents the trimeric form of the protein. The next figure will illustrate the allergic reactions that are involved, or the cellular mechanisms that are involved in the development of the peanut allergy. So in number 1, the first time a B cell is exposed to ARA H1 allergen, there is no allergic reaction that will occur, but plasma cells will initiate an overproduction of the IgE antibodies. The IgE molecules will attach themselves to mast cells or basophils. When the ARA-H1 enters the body for the second time, the IgE antibodies will react by binding to the allergen. The IgE-primed mast cells and the basophils will then release granules containing histamine and other allergic mediators. This will cause the allergic reaction. Another role of histamine is in inflammation. So histamine has a role in acute inflammation. In this figure, we have here the presence of an acute inflammation induced by the presence of a cut or a wound. In the presence of a cut or a wound, the mast cells that are present in the vicinity of the wound will detect the injury to nearby cells and release histamine, initiating an inflammatory response. In the next figure, in letter B, so after the histamine will be released, it will increase the blood flow to the wound site and increasing the vascular permeability. So it will increase the permeability of the capillaries, allowing the fluid and the proteins, as well as the phagocytes and other immune cells to enter the infected tissue. These events result in a swelling and reddening of the injured site and the increased blood flow. Increased blood flow to the injured site, causing it to feel warm. Inflammation is also associated with pain due to these events, stimulating the nerve pain receptors in the tissue. The interaction of the phagocytes with cellular distress signals and PAMPs and opsonins on the surface of the pathogens leads to the release of more pro-inflammatory chemicals, enhancing the inflammatory response. The next part will talk about the pharmacological effects of histamine. When we say pharmacological effect, that may be defined as the physiological and or biochemical changes in the body produced by a drug in therapeutic concentration. The first group of pharmacological effects that we are going to define are the effects on the vascular smooth muscle. When we say vascular smooth muscle, that is a type of smooth muscle making up most of the walls of the blood vessels. So vascular smooth muscle is found in the arteries, arterioles, veins, and lymphatic vessels. For the vascular smooth muscle, histamine induces intense vasodilation in all small blood vessels. So the vasodilation is mediated via the H1 and the H2 receptors. And this is manifested particularly in the capillary beds in all small blood vessels, including the arterioles, the capillaries, and the venules. In general, because there is vasodilation, so it can cause the development of a hypotensive crisis. However, histamine has a constrictive effect on large blood vessels, and its intensity is species dependent. We also have the phenomenon known as the histamine shock. So in histamine shock, this is brought about by intense dilatation of the capillary bed and is accompanied by increased capillary permeability. So the capillary endothelial cells constrict, exposing the endothelial membrane, which becomes freely permeable to plasma, lowering the blood volume. The dilated arterioles, venules, and capillaries tap large volumes of blood, reducing the venous return and the cardiac output. So this is another diagram showing the pharmacological effects of histamine on the small blood vessels, arterioles, capillaries, and venules. So generally, histamine will induce vasodilation. It will also increase the permeability of the blood vessels, resulting in the accumulation of fluid and edema. Because there is a decrease in the peripheral resistance, there will be vasodilation, and this will result in systemic hypotension. In humans, IV histamine can induce a fall in the blood pressure, cutaneous flushing, a rise in skin temperature, and intense headache. So on the heart, histamine has a direct stimulatory effect, both positive inotropic and chronotropic effect. When we say positive inotropic effect, this means increase in heart contractility. When we say positive chronotropic effect, this means increase in heart rate. So this is mediated via the stimulation of the beta-1 adrenergic receptors in the heart. The stimulation of the cardiac activity involves a direct action and reflex activation of the sympathetic nervous system, which is activated by the low blood pressure. There is also an increase in the coronary blood flow. In the intradermal tissue, intradermal injection of histamine produces a triple response that is similar to that first described by Lewis, and that was known as the triple response of Lewis. So this phenomenon was first described by Sir Thomas Lewis in the year 1924. Lewis triple response is the characteristic three-part response that develops when a line is made by a pointed object, such as a key on the skin. It is produced due to the release of histamine from the mast cells. For the pharmacological effect of histamine on the intradermal tissue, so it can cause the development of the triple response. Intradermal injection of 10 to 20 microgram of histamine produces a triple response. Insect and plant stings mimic many of these responses. The components of the triple response include, first, the red reaction. So during the red reaction, there is intense reddening at the point of injection, called the flush. That is due to the vasodilatation of the arterioles at the site of injection. After that, there is flare. Flare is brought about by the dilation of the arterioles that extends beyond the injection site, and this involves the axon reflex. The next part of the triple response is the wheal. Wheal means swelling. It occurs at the injection site due to the separation of the endothelial cells and edema caused by increased capillary permeability, which is due to the H1 receptor mediated contraction of the endothelial cells. After the formation of the triple response, the intradermal injection of histamine causes pain and itching by stimulation of the H1 receptor on the sensory nerve endings. The triple response due to intradermal histamine injection is based on the triple response of Lewis. This diagram shows the evolution of the inflammatory processes in the three parts described by Lewis. The triple response by Lewis consists of three aspects. When a line is made by a pointed object, these three events are likely to follow. First is the formation of the red spot. The red spot here is caused by capillary and arterial vasodilatation. After that, there is the formation of flare. Flare is a redness in the surrounding area due to arteriolar dilation mediated by axon reflex. It also refers to the irregularly outlined area of red skin spreading beyond the red line. The skin receptors in the epidermis are stimulated by the release of histamine from the mast cells. The third is the wheal formation, and this is caused by the exudation of the extracellular fluid from capillaries and venules due to histamine release. Also, it is referred to as the localized edema in the region of the red line. The red spot emerges within 15 seconds. The flare can take up to 45 seconds to begin, and the wheal can take up to 3 minutes to begin. So we have here another diagram showing the events after the intradermal injection of histamine. So there is, again, the triple response manifested by redness at the immediate injection site. There is also the development of flare, described as a larger red area brought about by the axon reflex, local axon reflex, and the wheal formation due to increased vessel or vascular permeability. After that, there is the development of pain and itching as effect of the stimulation of the nerves. For the GIP, particularly on the intestinal smooth muscle, histamine induces the contraction of the intestinal smooth muscles, causing an increase in the tone and motility of the intestine. For the species differences, the ileum of the guinea pig is considered to be most sensitive to this effect. Hence, it is used for the bioassay of histamine. Rats and mice are highly resistant to this effect. For the glandular tissue, histamine can stimulate the glandular tissue to increase secretion. A most important action of histamine is its ability to increase gastric acid and pepsin secretion from the gastric mucosa via the H2 receptors. For the summary of the pharmacological effects of histamine on the vascular smooth muscles, as well as the extravascular smooth muscles. So these are the effects of the histamine, particularly on the large blood vessels. So it will induce constriction of the large blood vessels. We also have for the smooth muscles that are present in the bronchi, the GIT as well as in the uterus, so generally there is contraction. For the summary of the pharmacological effects of histamine on the glandular tissue, particularly on the exocrine tissues or the exocrine glands. So these glands are generally stimulated to increase its secretion. For the central nervous system, injected histamine cannot cross the blood-brain barrier. For the endogenous histamine in the brain, it serves as a neurotransmitter function, and it can induce CNS excitation via the H1 receptor. Histamine, particularly endogenous histamine, also exerts autoinhibitory control of the synthesis and release of histamine, acting presynaptically via the H3 receptors. For the pharmacological effect of histamine on the peripheral nervous system, histamine causes itch and pain on the intracutaneous injection due to a stimulation of the sensory nerve endings. This diagram shows the mechanism of action of histamine on different receptors, particularly on the histamine receptors, in order to produce its effects in the body. So currently, there are 4 histamine receptors that have been identified. Now we have H1, H2, H3, and H4. When histamine will bind to the H1 receptors that are present in a particular tissue, such as the small blood vessels, such as the capillaries, it will induce the increase in the capillary permeability. And when it will bind to the H1 receptors that are present in the small blood vessels, it will also induce the vasodilation. And this is mediated via the mobilization of the calcium ions. When histamine will bind to the H1 receptors that are present in the sensory nerve endings, so it will produce pain and itching. When histamine will bind to the H2 receptors that are present in a particular tissue, such as the gastric parietal cells, it will mainly increase the gastric acid secretion, and this is mediated via the stimulation of the cyclic AMP. When histamine will bind to the H3 receptors, so it will induce the inhibition of the accumulation of the cyclic AMP, and it will result in the inhibition of the synthesis of histamine, as well as decrease in the secretion and the release of histamine. We also have here another diagram showing the mechanism of action of histamine. So histamine mediates its effects by interacting with the G protein coupled histamine receptor, H1, H2, H3, and H4 receptors. So the histamine receptors that are present in specific tissues are considered to be GPCR or G protein coupled receptors. The histamine will produce its effects by using or utilizing different types of G proteins, as well as utilizing different types of second messengers. We also have the IP3 or the inositol triphosphate and the diacylglycerol, as well as the CAMP or the cyclic AMP messengers. And this stimulation or inhibition of the second messenger, as well as the utilization of different enzymes, will produce its biological activities. So this diagram shows a summary of the histamine effects and the type of receptor that it utilizes in order to produce this particular effect. For the bronchial contraction, this is mainly mediated via the H1 receptor. For the large artery contraction, this is also mediated via the H1 receptor. For the microvessel dilation, So histamine will act directly on the blood vessels to dilate the arteries and the capillaries, and this is mediated via the both the H1 and the H2 receptors. In terms of the therapeutic uses of histamine, so it is said that histamine has no therapeutic application but only used for experimental pharmacology. Practical applications of histamine are limited to uses as a diagnostic agent. The histamine agonist, histamine phosphate, is used to assess the nonspecific bronchial hyperactivity in asthmatics and as a positive control injection during allergy skin testing. It can also be used for diagnostic purposes, for testing gastric acid secretion, and also for pheochromocytoma. However, its profound side effects limits its use. Another histamine agonist is the betasol. So betasol is an analog of histamine. It is an H2 receptor agonist, and it has a tenfold selectivity for stimulation of the gastric acid secretion over vasodilation. It is used in testing gastric acid secretagogue function in man.